Okay, thanks again very much for inviting me. I would like to focus on the peripheral nervous system controlling the heart, and just say when I was a graduate student, and I want to also mention how lucky I am to share the podium with two people I respect enormously this morning. This old-fashioned man, David, who's 10 times brighter than I can think of, and Bob Foreman, who was a graduate student with me in these ancient days when everyone thought about central neural command controlled the heart, the parasympathetic and the sympathetic nervous system, and Matt Levy came up. No.
Hold on real quick.
Thank you. Is that okay? Oops, sorry. And Matt Levy introduced the world of reciprocal innervation. What I would like to discuss is, like any other nervous system, talk about what you learned in medical school of afferents, efferents, and the interactions going on between the two. And so the heart and major vessels have enormous number of sensory neurites, as they're called, and they're concentrated very much in the outflow tract of the two ventricles. You notice in the papillary muscles, dorsal atrial, the areas you just love to burn, around the SA node, and of course, beautiful mechanoreceptors in the aortic arch. And you can inject chemicals into those tissues and label where neurons are located. In this instance, with Jeff doing DRG neurons, and then cross-correlate to see what possible chemicals those neurons can express. In this instance, nitric oxide synthase. NO is just a gorgeous activator of neurons of the intrinsic cardiac nervous system. Substance P, good old substance P. Everyone's hung up on substance P, I think, and they're incorrectly hung up on it. I haven't got time to talk about that. But in Jeff's lab, we've shown that you can have a brief occlusion of a coronary artery, and the neuron, the afferent neuron, goes on perhaps for hours. So you can think of angina, poor bugger. Hey, just going nuts centrally to your world, David. And then, this is a very superficial discussion, guys. The nodos, jumping over the nodos, that's another big afferent. And you can label those little guys up there from the heart. About 1% of neurons in the nodos go to the heart. And I was lucky enough recording a neuron for hours. And at some stage, the neuron began to burst, and suddenly the heart fibrillated. So these guys are very sensitive to what's going on inside the target organ. And here's a picture of an afferent in a mediastinal ganglion. This happens to be in the middle cervical ganglion. And this instance, occlusion and then reperfusion when it bursts. So that is my little story about afferents. Totally superficial, guys. But you have these different afferents in different stages of your thoracic system, central nervous system, all responding delicately to local things going on throughout your heart. So if I may, I'm going to switch to efferents in this superficial discussion. And you can label, you can inject into the heart and label neurons, and this happens to be a non-human primate showing the stellate ganglion. You can see the axons, you collect neurons there, which I will discuss a bit more at length. And for instance, in the canine model, the cardiac neurons in the stellate ganglion, I'm going to slightly focus on the stellate ganglion because of my clinical colleagues at this university are kind of hung up on the stellate ganglia. So the cranial pole, which is really the inferior cervical ganglion, is labeled with cardiac neurons, and these neurons are labeled from other parts of the body, the arm, the leg, all that kind of junk. And you'll notice they also can have peptides. Inside the stellate ganglion, there's every peptide that is found in your brain. And I just could focus on NPY. I just love VIP neurons. They're just so beautiful. And the way they're organized in rosettes, substance P, everyone's hung up on substance P, I know that. And if you inject, actually, substance P in a very small quantity in the right area of one of these ganglia in the thorax, you get enormous cardiac changes. So these are very powerful neurons dealing with really weird, in my estimation, but obviously not weird, chemical transmitters, non-normal transmitters. So we finally come to the human, and particularly at UCLA, they're focused on removing a portion, a specific portion of the stellate ganglion. You notice that previous anatomy. They're leaving the cardiac parts alone and just dealing with the controllers of the cardiac neurons. And here is, if you don't believe me, a nice stellate ganglion in a human on the other side. This is, of course, the left-hand side. And now I want to switch into the intrathoracic nervous system. And so you can take a ganglion, in this instance, a middle cervical ganglion, and you can chronically decentralize it and go in 2 weeks later and start to see what the neurons are doing. Now, if you believe in the fact that everything's from the spinal cord, there should be no neuron. Am I right? Good. Thank you, Mar. So here you have, in fact, beautiful activity going on in neurons in the MCG, chronically decentralized. And what is unusual for me, and I truly don't understand, but some of these neurons, which I will identify as local circuit neurons, do you see how they're specifically phase locked to the cardiac cycle? You have a little computer inside your thorax, and it's tracking each part of the cardiac cycle all your life. And you can, that's Bob Foreman's work, wherever he is. So you see in the human heart nerves coming down here. Look at that big nerve down the LAD. And so I would like to now introduce you to some concepts about where those nerves are going. And you recall that when I was a graduate student, those were all parasympathetic efferent postganglionic neurons. So the intrinsic cardiac nervous system now is found to have quite large ganglia, ganglionated plexi, we like to call them. And if you record the neurons in situ in those, this is work with Jeff, and he's got better hands than me. We did a chronic decentralized preparation and waited 2 weeks for everything to degenerate, so only neurons in the heart were functioning, and you could touch a part of the heart and get an afferent neuron to fire. Ergo, there are afferent neurons on your heart. There are also parasympathetic efferent. Everyone knows that. They learned that. But did I get in trouble talking about these guys? Sympathetic efferent postganglionic neurons are on your heart, and they're really important. So here's anatomy. Butyrylcholinesterase for you guys who think of Alzheimer's disease, which is slowly sneaking up with my age group, and, of course, acetylcholine. You inject microliter quantities beside an identified neuron in the right atrial ganglion plexus, microliter quantities, and you get beautiful forced depression in the ventricle. You can inject other neurons and get bradycardia. Hugely controversial, no longer, thank God, is the adrenergic neurons in the intrinsic cardiac nervous system. 2 different markers. This is done by our friend in Tennessee. This is human, taken out acutely at the OR. And then you can inject, again, a chemical into the intrinsic cardiac nervous system and get enormous sympathetic. So you have inside your heart afferents, sympathetic, efferent, parasympathetic efferents, but the vast majority of neurons are these. And these, you'll notice, never leave the ganglion. This is one ganglion doing confocal microscopy at multiple levels, so I can give you a 3D view of that whole ganglion. One ganglion, most of the neurons not leaving that ganglion. And if you record from those neurons, and I truly don't understand this, you can notice this one unit, you can see it picked up here, but look where it is in the cardiac cycle, always at one specific phase. So you have a computer on your heart that is somehow working out how every neuron should behave throughout your life, and it's sensing every part of your heart. How do you like that? And so with Jeff and various people, we've tried to correlate the whole population without bothering you with all the statistics, that if you record neurons on the heart, the vast majority of these local circuit neurons that are buried deep functionally within in an intellectual sense. You have ones that are coming in from the vagus and sympathetic, and there's ones that are transducing all the cardiodynamics. But in the middle of them all is a computational capacity that is not directly related to efferents or afferents. To me, I find that an intriguing group to think of targeting. And so if you're recording multiple neurons in one animal and looking at where they are in phase to the cardiac cycle or not, some are phase locked for the cardiac cycle and some are not. So they're computing different things, what is happening in the heart. And here, Jeff and I recorded neurons for 4 hours, 2 neurons for 4 hours, and looked at how phase related they were. And of course, I could have done them for years, and they'd stay phase related. Isn't that fun? Now, I'm going to be not nice now, because certain people get into the ball, they isn't that a nice little toy, the intrinsic cardiac nervous system? And some people say neurons in one area project to one part of the heart, and so you can burn it, and ergo, no arrhythmias or no whatever. You know, God did not make the brain and the heart like that. Unfortunately, every single collection of ganglion plexi innervate the SA node. And if they're screwed up a bit, you can get atrial tachycardic arrhythmias by putting microliter quantities of a chemical beside the right neuron. So it's an easily screwed up system. And just to throw you off, this is the right atrium. You guys know all about that, SA node. But I was activating with microliter quantities a ventricular neuron. So ventricular neurons control all over, and so do atrial neurons. And just finally, I would look at ischemia. So if you notice the ganglion plexus and the atria, say in the ventricle, they have multiple vascular supply. And in this instance, most of them come from two. This is from the other side, this is from this side. So most are getting blood supply from multiple areas. So if you have trouble with the blood supply, it will survive, correct? However, in a porcine model, if you obstruct the artery perfusing the neurons you're recording from, they get excited like heck, and it will last for long periods of time, just like Jeff's stuff with the afferents, so will the intrinsics. And so it's a long duration effect. And with my good colleague Dave Murphy in Halifax, we were able to extract ganglion plexus from people undergoing bypass surgery. And if you look at the ganglion plexus targeted by the diseased artery, if that makes sense to you, please look at this. This is a normal, and this is an abnormal. Isn't that terrible, what the disease process of the intrinsic cardiac nervous system? And if you don't buy that, look at the EM of a neuron. Would you be happy to have one of those running around for your heart? Now, a controversial subject, which I love, is, with apologies to some people in the room, nerves over infarct. Some people think that a nerve over an infarct kills the nervous system and creates all kinds of havoc, forgetting the fact that every nerve in your body carries its own blood supply. They are unaffected by infarcts. However, if you go down, and this is a guinea pig, so you get the whole infarct under the microscope, and you look at what happens, and we were trying to put progenitor cells in the infarct and look at all that, that failed because there's no blood supply. But guess what we found? Was on the edge of the infarct, flowing into the infarct, first comes the neurites. Please remember, the neurites are fed distally from wherever you are. That's why nerves over infarcts are not touched. The cell bodies are fine. So in an infarct, they invade the infarct with these axons. The axons bring blood vessels with them, and then muscles follow. So after about 3 or 4 weeks, a huge, because it was volume I'm not sure of, but on a microscope it was half reduced. So my only point, people, is that maybe it would be nice to think of neurons. This is the last thing that we also had a stab, at recording neurons in the intrinsic cardiac nervous system in humans undergoing surgery. And guess what? You could touch the ventricle and activate neurons. So there is a tremendous opportunity, I would think, in the future to look at this nervous system And before I quit, I want to say how grateful I am for Pradeep and Ken, Una, Ray, David, and Eileen for helping me at UCLA. Young people full of tolerance of a somewhat slow old man. Thank you.